welcome to the Chats with Pratt's channel. So last video in the germination of seed, we learned about the types of seeds, and now we are going to study about the bean, specifically bean seed. So bean seed has seed coat, it has hilum, micropyle, embryo, then embryos, division, mesocortal, epicortal, plumule, hypocortal, radicle. So we'll learn all these things in brief as well as in detail. Okay, now let's get started with the shape of the bean. So the shape of the bean is like concave and convex, and it is dicotyledonous and exalbuminous. In the last video, if you guys have seen, exalbuminous is nothing but a seed without endosperm. So bean seed is a seed without endosperm, and it is dicot. That means it has two cotyledons. And then it is made up of three main parts, which is seed coat, embryo, and cotyledons. So the seed coat is the protective covering. The seed coat's function is to protect the inner part of the seed, and the seed coat has two layers. The outer layer is called tegmen and the inner layer is called testa, or vice versa. Yeah, so the outer layer is called testa and the inner layer is called tegmen. The outer layer is the brownish part, hard part, which protects the seed from the any natural processes or insects, etc. And tegmen is the inner thin white layer that is usually fused with testa, and it protects the inner part of the seed. So testa is the outer part of the seed coat, tegmen is the inner part of the seed coat. Testa is brownish in color, tegmen is white in color, and it also protects the inner part of the seed. Now we will study about a distinct scar caused by the detachment from the placenta. So this is called hilum. Hilum is a distant scar on the concave side of the seed. That means this side of the seed, a concave side. It is place where the ovule was attached to the placenta of the ovary. Ovule and ovary you guys must have learned in the previous lesson. With a short stalk called funiculus. So funiculus connects the ovule with the placenta, and when the seed is matured, then the funicles is detached, and the area where the funicle was connected with placenta is this distinct scar. So in bean seed, you guys must have observed there will be a distinct scar. That means a different color of a scar. So for example, let's take bean. So bean is reddish color and it has something white in between those reddish parts. So that is hilum, and then adjacent to a hilum is micropyle. Micropyle is in the previous lesson we learned about the micropyle, which is from which the respiration of gases takes place, and helps in the diffusion of the respiratory gases for the growing embryo. Then below the seed coat are two thick cotyledons of the bean seed, and they contain the food reserves, as it is exalbuminous. That means they do not contain endosperm at all. So cotyledons contains the food material, that's why it is fleshy, it is thick. And then we will speak about embryo. When we speak about embryo is a miniature plant. So it also has a few different parts or stages. So it consists of two major parts, that is radicle and plumule. Radicle is the shoot, is the root part, and the plumule is the shoot part. So plumule is the shoot part which grows into the stem, and radicle is the root part, root system, which when gets matured will grow into roots. And the axis between these two plumule as well as radicle is known as embryo axis. So the axis between plumule and radicle is known as embryo axis. So the embryo axis is differentiated into 5 parts. So the 5 parts is mesocortical, epicortical, hypocortical, radicle and plumule. We have learnt about radicle and plumule. Radicle forms the root system, plumule forms the shoot system. Mesocortical, now the mesocortical is the part of the embryo axis where cotyledons are attached laterally. So when we talk about cotyledons, they are attached laterally to the embryo axis. So this is where mesocortical is this is where mesocortical is this is where mesocortical exists. Epicortical is the portion between the embryo axis. Epicortical is the portion of the embryo axis between mesocortical and plumule, and hypocortical is the part of the embryo axis which lies between mesocortical and radicle, which forms the root system, whereas epicortical lies in between the mesocortical and the plumule, which forms the shoot system. And then we have learned about the plumule and the radicle. Now we are done with the bean seed. Okay, now they have given us about the maize seed. Okay, so maize seed is one-seeded fruit. That means it is monocot, and it has endosperm in it, so it is albuminous as well. So it's monocot and it is albuminous. What is monocot and albuminous? The maize seed. Now bean seed is done. We are done with that chapter. Now we will come on to the maize seed. So maize seed is monocotyledonous, and it is albuminous. So in maize seed there is one phenomena which takes place. So what happens is, the fruit wall, which scientifically is called as pericarp. So the pericarp is fused with the seed coat to form a single layer. Now this phenomena is called caryopsis. What is this phenomena called as? Caryopsis. The fusion of the fruit wall and the seed coat is known as caryopsis, and the fruit wall is known as pericarp. Is that clear to everyone? Okay. Now this type of seed is known as grain. We call it as grain, maize grain. Okay. So mostly monocot seeds have the yellow area, the endosperm, where the outer layer of the endosperm. So the outer layer of endosperm is known as alluron layer, which is like yellowish color, straw colored, and then the endosperm is also yellowish. And when we are talking about this shape and the size of the maize seed, so the maize seed is flat and triangular. Okay, and the endosperm is rich in starch, and the layer which I spoke about, the alluron layer, which is the outer part of the endosperm, is rich in protein. Endosperm is rich in starch, alluron layer is rich in protein. Is that clear to everyone? Okay. If it is not clear, okay, rewind the video and then pause it until you guys understand it. Okay. Okay, now, so it is given here that hilum, which is this distinct scar which we spoke about in the bean seed, and the micropyle, which is adjacent to the hilum, is not distinguished as in the bean seed. So in maize seed, it is not distinguishable. Okay, and the embryo lies in the lower part, whereas uh, the embryo lies in the lower part, the endosperm lies in the upper part. Okay, so the embryo lies in the lower part of the seed, the endosperm lies in the upper part of the seed, and the single cotyledon, which is monocot, is also known as cutilum. We call it as cutilum. So the monocot seed, the one cotyledon seed, monocot, there is no difference, guys. They are just given us that it is cutilum. Okay, you can call it monocot, you can call it cutilum. It is same thing, like two words and one meaning. That's it. And the embryo is also comprised of plumule, which is the growing shoot area, and the radicle, which when becomes mature turns into root system. So now those plumule also has the outer covering called cleoptile. Okay, so cleoptile is the protective layer of the plumule to avoid any further damage by the natural agents such as wind, water, insects, etc. Okay, so plumule is enclosed in a protective sheath called cleoptile. Again, it is only possible in monocot and albuminous seeds, monocot as well as albuminous. These two features should be there in the this function to take place. Not necessarily albuminous, but it should be monocot. Okay, and then the radicle. The radicle also has the protective covering sheath, which is known as coleorhiza. So it sounds similar, isn't it? So coleoptile is for plumule, coleorhiza is for radicle. You guys can remember it like that. So it's coleoptile, coleorhiza. Coleoptile is for It is P is there, so plumule, and coleorhiza R is there, so radical. Is that clear to everyone? Okay. Now we will see the difference between the bean seed, which we spoke about in the just few minutes ago, and maize seed, which we just now spoke about. So bean seed is dicot. It is fleshy. It is exalbuminous. That means it does not have endosperm, and then the embryo is large. The seed coat is thin and brownish in color. Hilum and micropyle are visible. Tips of plumule and radicle are naked. That means they do not have coleoptile and coleorhiza. And then the reserve food is carbohydrates and proteins, which is present in the cotyledons. And seed is separately contained in the fruit. This is in bean seed. Now in maize seed, it is monocotyledon, it is albuminous. That means endosperm is present, and then the embryo is small, and the seed coat is very thin and fused with pericarp. This phenomena is known as caryopsis. Then the hilum and micropyle are invisible in maize grain. Then the tips of plumule and radicle are covered by coleoptile and coleorhiza respectively. Then the reserve food material is starch, and the seed wall and the fruit wall are fused to form a grain. They are basically repeated. The seed coat is very thin and fused with pericarp, and the seed wall and the fruit wall are fused to form a grain. Extra point, what to do? Study. Now, differences between the monocot and the dicot seeds. Bean seed, maize seed, when we have already studied about monocot and dicot. Why do we need to study again and again? So, this is because we need to get better clarity on monocot seed and dicot seed. Maize grain and bean seed was just an example of monocot and dicot seed. Monocot seed has single cotyledon. Everybody knows. Now at least you guys know, isn't it? Don't tell me you guys don't know. This was told in the first part of the video. Then the dicot seed has 2 cotyledons. And the monocot seed, now I'll only read out the monocot seed differences. The monocot seed is the food found in monocot seed is stored in endosperm. Okay, and then the alluron layer, which is protein rich, which has protein rich cells, is present outside the endosperm. That means it is the outer layer of the endosperm. Then the embryo tips may bear special sheets of coleoptile, of plumule, and coleorhiza of radicle. And then the embryo occupies one side of the seed, and the plumule lies at one end near the cotyledons. So these are the 6 differences of monocot seed. Now dicot seed, the food may be stored at endosperm or cotyledons. There are options given here. But in monocot seed, the food is stored only in endosperm, and there is an exception in orchids. Orchids, it can be stored in both endosperm and cotyledons. Yes. Third point is that the alluron layer is absent in dicot seed. The alluron layer, which is